Hello, we are representing Sam Houston Forensic Science Department. Today we will be discussing DNA serology, specifically how biological samples are collected, packaged, and analyzed in the crime lab.
Before we begin, let's talk about why DNA evidence is so important. DNA can uniquely identify an individual, except for identical twins, which makes it one of the most powerful tools in forensic science. It can confirm the presence of a person at a crime scene, link multiple crimes together, or even exonerate someone who has been wrongfully accused.
DNA serology is the process of identifying and testing biological fluids like blood, semen, and saliva before full DNA profiling is done in the lab. It's often the first step in DNA analysis, and it plays a crucial role in determining which samples are suitable for further testing.
DNA can be found in a variety of biological fluids and materials. The most common sources include blood, semen, saliva, urine, and touch DNA. Each of these samples must be handled carefully to protect the DNA from contamination and degradation. Now let's look at how to collect the most common DNA containing samples. For liquid blood, use a sterile swab and allow it to air dry. For dried blood, use a moist and sterile swab, then air dry it completely. Semen is often found on clothing or bedding. Use an alternative light source like UV or blue light to locate the stain, then swab the area carefully. Saliva might be found on items like cigarette butts, cans, or bite marks. Use a moist and sterile swab and rotate it on the surface to collect the cells. For surfaces like weapons or door handles, use the double swab technique. First a moistened swab, then a dry one. This helps lift both visible and invisible DNA residues.
Maintaining a clear chain of custody is just as important as the evidence itself. Anytime the evidence is transferred from crime scene to lab or between personnel, it must be recorded. The log should include the case number, date and time of collection, collector's name and initials, and the description and location of the evidence. A broken or undocumented chain of custody could mean the evidence gets thrown out in court.
Whether you're at a crime scene or in the lab, proper protective equipment is essential. It protects you from biological hazards and ensures you don't contaminate the evidence. This will include your lab coat, gloves, and a mask. An alternative light source is used to detect and visualize stains on evidence. This gives the analyst an idea of what other tests to perform. Blood will absorb the light, which causes the stain to appear darker, while saliva and semen will fluoresce under the ALS.
The Kastle-Meyer or phenolphthalein test is a method used to detect hemoglobin. If hemoglobin from blood is present, it speeds up this reaction, turning the colorless phenolphthalein into pink phenolphthalein. A positive result will turn pink in color within 30 seconds. This is a non-human specific presumptive test, meaning it will need to be confirmed with a more specific technique.
To perform the test, cut a small piece of the cloth, then apply 2 to 3 drops of 70% ethanol, followed by 3 to 4 drops of phenolphthalein reagent, and finally 3 to 4 drops of hydrogen peroxide. A pink color appearing within 30 seconds indicates a positive result.
If the Kastle-Meyer phenolphthalein test is positive, it is then followed up with the ABA card hemotrace test, which is human specific. The ABA card hemotrace is an immunochromatographic test that is capable of detecting trace levels of human hemoglobin. A positive result will show 2 pink lines indicating that the hemoglobin level is at or above 0.05 micrograms per milliliter. A negative result will show only 1 pink line in the control area.
To begin testing, cut a sample from the stained cloth and place directly into the provided extraction buffer and incubate it at room temperature for 5 minutes. After incubation, remove the test card from the sealed pouch and label it. Add 4 to 5 drops of the extracted sample into the sample well marked S on the card. Results should be read at 10 minutes, though a positive result may appear as early as 2 minutes. A positive result is shown by 2 pink lines, one in the test area T and one in the control area C.
The acid phosphatase test targets an enzyme called acid phosphatase, which is found in large amounts in seminal fluid. When the sample is mixed with chemicals from this test, a purple color will appear if semen is present. The results are given in a score ranging from 1 to 4, 1 being a slow pink color develops on a stained material, and 4 being the intense color develops and bleeds. This is also another presumptive test.
To perform the AP test, begin by moistening filter paper with DI water or using a pre-wetted swab. Using a transfer pipette, apply 2 to 3 drops of solution A, followed by 2 to 3 drops of solution B. A positive reaction is indicated by a purple color developing within 10 minutes.
The ABA card P30 is a test that detects the prostate-specific antigen P30, which is a protein found in semen. If P30 is present, it will bind to the antibodies found in the card, resulting in a pink line. A positive result will show 2 pink lines indicating that the P30 level is at or above 4 nanograms per milliliter. A negative result will show only 1 pink line in the control area.
To perform the P30 test, cut a sample from the suspected stain and place it directly into 750 microliters of the provided extraction buffer. Incubate at room temperature for 30 minutes, then briefly centrifuge. Remove the test card from the sealed pouch and label it. Using the supply dropper, add 6 to 7 drops of the extracted sample into the sample well marked S. Read the results at 10 minutes, though a positive result can appear as early as 2 minutes.
Serology is used to detect trace amounts of saliva. It targets amylase, which is an enzyme found in saliva. If saliva is present in the sample, the colorless solution will turn yellow. If saliva is not present in the sample, the solution will remain colorless.
To perform the Serology test, extract half of the swab in 50 microliters of water and incubate at room temperature for 30 minutes. Once the extract is ready, add 8 microliters of the sample to the Serology solution and mix gently to ensure it is well combined. Read the results after 10 minutes, though a positive result may appear as early as 30 seconds. A yellow color change indicates a positive result, while no color change indicates a negative result.
The Christmas tree stain is used as a confirmatory test for semen. Spermatoza detected in a stain or on a swab confirm the presence of semen. For spermatoza examination, material from the stain or swab is fixed to a microscope slide, stained, and examined under the microscope for cells with spermatoza characteristics. This procedure uses nuclear fast red and picro indigo carmine. This stain combination is called Christmas tree because the heads of the sperm are stained red and the tails are stained green. After staining, the sample is viewed under a compound light microscope.
The serology testing is now complete. We've identified the presence of biological materials suitable for DNA analysis. Next, we'll begin the DNA extraction process.